Okay, good afternoon, everybody, and welcome back. Just before we continue, I just want to emphasize that you have a test. Your next class will comprise a test for the whole period, okay? And it's multiple choice. All you do is go to Canvas and go to the quizzes section. Just hit the quizzes button and you'll see the test light up. You just open it and you do it once you've begun. You'll have the whole class period completed, but it must be completed during the class period, during the set class period, that is 4:05, from 4:05 to 5:30. All right. And it is up till the end of our last class. So that is the metabolism slide set 1. So up to the end of the metabolism slide set 1. Although I will be covering 1 or 2 of the slides, the last slides from that slide set again today. Okay, but study up until the end of the last class. All right. So with that said, let's continue with our discussion about metabolism. But before we do, I just want to go over again something that I explained rather badly last time. And that is this question of enzymes functioning together with other components. And the first set of components that we need to think about is the enzyme itself, which is the vast majority of enzymes are proteins. So an enzyme comprised of a very complex protein structure of one sort or another. But many enzymes do not function when they are simply the protein on its own. Some do, but many of them, the protein itself is not functional until it is combined with something else, which is a non-protein. And that we refer to as a cofactor. So the cofactor is something, a component that combines with the protein portion of an enzyme to make it active. The protein on its own, we refer to as the apoenzyme. There it is there, and it is inactive. So that is the apoenzyme. It must combine with a cofactor of some sort. Very often, those cofactors are organic. And when they are organic, we refer to them as coenzymes. So many of the coenzymes, for example, are derived from the vitamins. And here are a whole set, the nicotinamide adenine dinucleotide, which we're going to hear about often, NAD+, nicotinamide adenine dinucleotide phosphate, NADP+, flavine adenine dinucleotide. These are usually known by their acronyms, NAD+, NADP+, FAD, and another one called coenzyme A, which we're going to hear about today again. So these very frequently will combine together with the enzyme. Again, when a cofactor is organic, we refer to it as a coenzyme. There it is here, combining here. Once it's combined, the two together form the holoenzyme. The holoenzyme means the entire enzyme. And the entire enzyme now is active. And these cofactors, look what these ones here are doing. They are all assisting with the transfer of electrons, of energy from hydrogen. That is what these two nicotinamide, you see this nicotinamide here. That means that many of our vitamin Bs, for example, are nicotinamide, form the basis for these electron carriers, which are coenzymes. There are some other cofactors which are not organic, and very often those are metal ions, either iron Fe in hemoglobin, ion itself, very often magnesium, things like this. But again, they are there to interact with electrons very often. Very often also these enzymes actually serve to switch, in a way, switch on and switch off the enzyme, because when they're bound to form the holoenzyme, the enzyme becomes active. When they are not, the enzyme is inactive. Okay, so I hope that clears it up. If just read through this carefully, and it explains a lot of that to you. Now, let's go back to something that we did discuss at the end of our last class. And I'll just briefly discuss it again because it's really important. And that is the fact that enzymes can actually interact with other substances in a way which will affect the action of their active site. So the first that we talked about was competitive inhibition. Competitive inhibition, in a way, is an artificial situation. It's a situation where an enzyme interacts with what is effectively a toxin, and the toxin binds to part of the active site and blocks it. That binding is almost invariably a permanent binding, and the competitor, whatever the substance is, cannot be unbound from the enzyme. So there's no system of control here. It's just that this toxin is actually blocking the activity of the enzyme entirely. I just want to try and do something. Just 1 second, please. Okay. It didn't work. All right. So that's competitive inhibition. Competitive inhibition can be things which, toxins which an organism might eat, for example. But also very often these are toxins which are produced, for example, by bacteria. Very often bacteria, like things which cause diphtheria, tetanus, all of these things are producing toxins which bind to enzymes and inhibit them. Systems of non-competitive inhibition, however, are different. Now, in non-competitive inhibition, let's have a look at this enzyme. Here's the whole protein structure of the enzyme here. Here's its active site there. This is functioning to bind the substrate. In order for the active site to bind the substrate, it has to match the shape exactly. And any distortion of that shape is going to stop the enzyme from being able to bind the substrate and act on it. These enzymes, which are designed to be controlled by non-competitive inhibition, have another site. It is not an active site. It's a site remote from the active site. Here it is here. And it's called the allosteric site. There's the word there, allosteric. And the allosteric site binds some other substance. It doesn't bind the substrate. It binds some other substance. And when it binds that substance, the whole shape of the enzyme changes and the shape of the active site changes. And it can no longer bind to the substrate and act on it. This gives the cell an exquisitely sensitive way of controlling the activity of an enzyme. Because if it binds this non-competitive allosteric substance to the allosteric site, then the enzyme can't operate. But this binding is not permanent. It's in fact quite transient. The substance will come in and out of there. Switch on, switch off, switch on, switch off, switch on, switch off the enzyme. So how is this used in the cell? Well, it's used in especially in a system known as feedback inhibition. And this is a system whereby a substance that is being produced by the cell actually goes back and acts as an allosteric substance. So let's have a look here. This is a metabolic pathway. Here's the substrate 1. Okay, we want to end up with an end product of some sort. Here's this initial substrate. The substrate binds to the first enzyme and produces a product. that product is different to the original substrate, but it produces an intermediate product. That intermediate product is bound by a second enzyme and produces this intermediate B. Intermediate B is bound by a third enzyme, which results in the production of an end product of some sort. So what very often happens is that this end product here acts as an allosteric substance on the very first enzyme. So what is happening? Well, the cell, let's just assume here, in the cell, there's very little of this end product, which the cell desperately needs. Okay, so the cell needs to make it. So in that situation, there's none of the end product. There's nothing to bind to the allosteric site. The substrate binds, the whole pathway runs, and the cell begins to produce the end product. Now, the concentration initially of the end product is very low. So a little bit might bind here at this allosteric site. A little bit might bind. So the enzyme might be switched off just occasionally, right? But as the concentration of this end product increases and increases and increases, so this enzyme spends more and more time bound to the allosteric substance and inactive until it's inactivated completely. It's just not operating because the concentration of the end product is so high. But remember what's happening. The cell is manufacturing the end product for a purpose. The cell is actually using the end product somewhere else. It's using it for something. So the concentration begins to drop. And as the concentration drops, so the enzyme begins to act more and more and more often. So what's happening is that the actual concentration of the end product is controlling the activity of the very first enzyme. And because it's controlling the very first enzyme, it's controlling the whole pathway. It's switching the pathway on and off in response to the concentration of the end product. This means it's not wasting energy. It's not wasting energy. It's not producing more than it needs. It's producing exactly the right amount, exactly the amount it needs. And so this system is called feedback inhibition, and it is very, very common. It's very common in any of the pathways that we're going to talk about, that the very first enzyme is sensitive to the concentration of the end product of the whole metabolic pathway. And the first enzyme acts as the switch. Why the first enzyme? Why do this at the first enzyme? Why not use one of these? Why aren't these ones, these two other enzymes, why aren't they allosteric as well, allosterically controlled? Because it would just be a waste. If this was not allosterically controlled, but the second one was, you'd produce all of this intermediates before you switch the pathway off. And it would just, this is not what the cell needs. Okay, the cell doesn't necessarily need this. It needs the end product. So if you want to switch the pathway on and off most efficiently, switch it off to the very first one. You switch off the first enzyme, you shut down the entire pathway. You don't waste, the cell doesn't waste time and energy manufacturing useless intermediates. It switches the pathway on and off at the very first enzyme. This is a typical structure of a feedback inhibition pathway. Okay, so the last thing I just remind you of, I always tell you when I talk about enzymes, I say most enzymes in the cell are proteins, but there is another set of enzymes that are extremely important, and those are the enzymes which are composed of RNA, ribonucleic acid. And ribonucleic acid enzymes are referred to as ribozymes. The name tells you everything that they do. It's RNA that acts as, that has enzymatic ability. Now, the very first we're not going to talk about them in any great detail, just remember this, that ribozymes can do various things in the cell. And very often they're extremely important in controlling other types of RNA. So, for example, they cut and splice RNA. We're going to learn later on that in eukaryotes, messenger RNA, for example, needs to be cut up and edited in eukaryotes before it goes on to be translated. That is done by ribozymes. There are various other places in the cell, especially in the manufacturing of RNA and of protein, where ribozymes act. But the one really important place, and the one that we're going to hear the most about, is in the ribosome itself. Ribosomes are composed of two things. They're composed of protein and they're composed of RNA, of structural RNA, which also has enzymatic ability. So the RNA, the ribosomal RNA that makes up the ribosome, also has ribozyme activity and is one of the most significant things in the process of translation. It's ribozyme that actually starts to add amino acids together and form peptide bonds. Okay, so that aside. So that, by the way, up to there, that's where your test will be on Wednesday. All right, so let's carry on with our discussion about metabolism in general. There are a few basic principles that I'm going to talk about and remind you of. You've done this in chemistry, I'm sure, but it bears repeating. Many of the reactions that we are going to be talking about are redox reactions. That is, they consist of a coupled oxidation and reduction reaction. And remember the definitions of those. Oxidation refers to the removal of electrons from an element or a compound. If we oxidize it, we're actually removing electrons. If a substance gains electrons, that is referred to as reduction. And the two are usually coupled together. So here we have, for example, a system where we have substance A, we're going to remove an electron from substance A, we're going to oxidize it. And we're going to add that electron to substance B. Substance B gains that electron, it is reduced. Substance A lost the electron, donated the electron, it is oxidized. So the two reactions, the reduction reaction and the oxidation reaction, we refer to these as the half reactions, and together they form the entire redox reaction. We are not going to go into all of the energetics. That is reserved for chemistry. But just remember that there are these two half reactions, one in which we have a donor, an electron donor, and that is the substance which is oxidized. And we have an electron acceptor, which is the substance that is reduced in the redox reactions. So we are going to watch this being done under special circumstances in biological systems. Electrons do not simply fly around the cell like little lightning bolts. Instead, they have carefully shepherded, and always as much energy as possible is preserved. But remember, all these reactions are inefficient to a certain extent. One of the laws of thermodynamics. We cannot have any energy transformation which is 100% efficient. So we are always losing some of it, but energetic systems in biology do their very best to harvest as much energy as they possibly can from these very, very energetic particles, which are the hydrogen electrons. And the point about hydrogen is that it is very easy to strip the electron off the hydrogen atom and have it participate in other redox reactions. Now, ultimately, we can't have just loose electrons flying around. Ultimately, they must be joined again to the original hydrogen atom. But in the meantime, in the period of separation, we can use those electrons, because as we pass them through a series of redox reactions, we can, at each step, the biological system will harvest a certain amount of energy to do work. We'll see how that occurs in a while. So many biological oxidation, most biological oxidations are dehydrogenation reactions. In other words, stripping hydrogen off, separating the proton from the electron, and using both. We'll see that biological systems actually use both of them. The electrons, the proton itself can be accumulated in solution. And the more protons you have, of course, the more acid a solution becomes, because that's the definition of an acid. An acid is something that donates protons to solution. But the electron can be harvested, and it can be actually transported together with its energy. And it is transported very often by coenzymes. As we saw before, these substances like nicotinamide adenine dinucleotide are actually able to take up electrons and temporarily at least hold them and transport them and donate them to another substance, either an enzyme or to a substrate or whatever. So the entire hydrogen molecule, H2, contains 2 protons and 2 electrons. Okay, so you can have a look here, and we're going to see this is some kind of organic molecule. Let's just think, for example, that it might be a fat or something like that, glycogen, something which has carbon with lots of hydrogens, and those hydrogens can be stripped off. And this substance here, NAD+, that is the same substance that we heard about earlier. Here, at our very first slide. Here it is here. Here they are here, if you want all the names and everything. So here's NAD+. It's an ion. The plus tells you that it is an ion. And it is this very powerful electron carrier that we're going to see quite often. And what is going to happen is, by enzymatic activity, we are going to strip off an entire hydrogen, H2, from our fuel source. And here it goes here. They're going to go to this nicotinamide adenine dinucleotide plus, and it is going to pick up 2 of the electrons. One of them is going to cancel out, is going to be cancelled out, of course. So we're going to see that it's going to, the charges and everything are going to change. It gets converted, it's being reduced, and it gets converted to this substance, NADH. What is that? Well, it's NAD plus 2 electrons plus a proton, and it releases a proton into solution. So it is NAD, that reaction is our fuel source, plus NAD plus, ends up with an oxidized organic molecule, and this substance, NADH, and a proton. All of this is very useful. This is now our electron carrier. It's carrying actually 2 electrons and all of their attendant energy. And it has released into solution this proton. We're going to see that the cell is going to make use of that proton, or this accumulation of protons. It's itself extremely useful to the cell in several ways. So there are several substances inside the cell which are able to perform similar reactions. They're able to pick up electrons and protons from organic substances and deal with them, manipulate them, and temporarily at least, they will transport those together with their energy to some other reaction that requires energy and requires electrons to do the reaction. The other substance, of course, that we need to talk about is ATP, because ATP, in fact, is the most useful source of energy that directly funds metabolic reactions. And this is we're going to find out that, in fact, all the energy that is being transported around by the electron carriers, that energy is going to be used to actually produce ATP. And ATP is synthesized by taking the substance ADP, adenosine diphosphate, which is a nucleotide, and adding a phosphate to it. We add here, there's the phosphate group there, and it's this bond here, which is particularly biologically useful. It's very unstable, but it's in the very instability allows it to be easily and rapidly used. And the energy is usually transported from ADP to some other substance, often to an enzyme, which then adds the energy to a substrate and does something, or some similar process whereby that phosphate together with the energetic bond is stripped off the ADP and is added to another substance to phosphorylate it. When that substance is phosphorylated, it's not the phosphate so much that's important. It's the energy associated with that bond that is really important. And so what we can visualize this. We have energy being transported around the cell in one form or another. We're going to temporarily add that energy in as a phosphate bond here to ADP and make ATP. That ATP is the most useful source of energy for metabolic reactions because this phosphate bond here can be transferred into a set of reactions together with all of its energy. We're left then with ADP again, and to make more, ADP is relatively stable, and it is used as the substrate to make more ATP. All the way along here, we're going to have to be feeding in masses of energy. And those are the processes really that we're going to be talking about from now on for a little while. So first of all, remember and understand this term phosphorylation. That when phosphorylation occurs, we're transferring a phosphate group, but what's really important is we're transferring the energy associated with that phosphate group. The other thing to say is, of course, the ATP is very unstable. This is a short-term solution. ATP is used very soon after it is produced. And if we want to store energy for a very long time, then we have to use the ATP to actually manufacture stable molecules with chemical bonds that can be accessed much later. And those are our storage products: starch, fat, lipids, all of these sort of things. And in eukaryotes, glycogen and substances like that we put into the muscle and into liver. Okay, so there are in fact 3 ways in biological systems that ATP can be made. And these are a substrate, they're called substrate level phosphorylation, which is basically a simple chemical reaction driven by enzymes. Oxidative phosphorylation, which is a complex membrane-associated process, which uses oxygen, which we're going to talk about extensively. And then photophosphorylation. This is one of the steps in photosynthesis. And we're going to see that all that is happening is that organisms which can use sunlight in order to change the energy of light into chemical energy those we call the autotrophs. They are able to take the sunlight, change the light energy to energy of a chemical bond, and they do one of the steps in the whole process is that they manufacture ATP using sunlight, using light rather, whatever the light source is. So substrate level phosphorylation is really like a chemical set of chemical reactions. And so we'll have some substance that has phosphates on it, for example, has a phosphate associated with it. And then ADP, adenosine diphosphate. And then enzymatically, what we're going to do is strip off this phosphate and add it to the ADP. So this can be done using fuels. And we're going to see an example in a while in the process of glycolysis, whereby we're going to strip off phosphates from substances. We're going to add them to ADP, and we're going to produce ATP. So never mind about the names here. All that I want you to notice. This will appear again in a little while. Again, you don't need to remember the name, but all I want you to notice is this is called 1,3-bisphosphoglycerate. Here it is here. And all that you need to notice is that it has phosphate groups associated with it. Okay. And that is put together with an enzyme called phosphoglycerate kinase. A kinase moves functional groups. And in this case, the functional group it moves is the phosphate group. And it moves the phosphate group from this to ADP, creates ATP, and leaves us with this substance here, 3-phosphoglycerate. Still got one phosphate there, which we need to keep an eye on. But it's lost a phosphate, which has been transferred. This is typical substrate level phosphorylation. It's a simple chemical reaction, enzymatically controlled. We'll see several of these in a while. The second one, we're not going to be discussing today, but we'll discuss at some length in our next class. And this is oxidative phosphorylation. And never mind about all of the details. I just want to point out a couple of things about this process to you. First of all, the whole aim of this complex set of reactions that you see taking place here is to produce ATP. And we're going to start off, have a look here. Have a look there. What is this? NADH. That is our reduced NAD+. It's been reduced and is carrying 2 electrons with it, right? That, in this whole complex system, what we're going to do is we're going to strip off those electrons, and we're going to send them through a whole series here of oxidation reduction reactions. Each of these substances here can pick up the electrons and then pass them to the next substance, 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 to the next, to the next. These are all oxidation reduction reactions. And as the electrons flow through there, they are losing energy, but the energy, as much as possible, is being harvested at several points to do some work. The nature of that work we'll discuss later, okay? But the end product of all of these reactions is the production of ATP. But in order for this whole system to work, we've got to catch these electrons at the very end. We have to somehow unite them with protons at the end of the whole process. And in this system, oxidative phosphorylation, which we also refer to as cellular respiration, we actually use oxygen. We use oxygen as the terminal electron acceptor. In fact, oxygen is sitting at the end of this whole series of reactions, like saying, come to mommy, come to mommy, come to mommy. It's pulling these electrons. It's pulling the electrons off this electron carrier, pulling them through all of these reactions, getting them to do work as they go. At the end, we're going to unite our electrons, 2 electrons, 2 protons, and an oxygen atom. And what we end up producing is H2O, water. So breathe out for me. And what are you breathing out? Sure, you're breathing out carbon dioxide, but you're also breathing out water. And water is a byproduct of cellular respiration. It's a byproduct of oxidative phosphorylation reactions, all of which are designed to produce ATP. And this, in fact, is the cell's most efficient way of producing ATP. Just remember the basics for the moment. Electrons going through oxidation reduction reactions, ultimately binding to oxygen, and in the process producing ATP. So that's oxidative phosphorylation. Oxidative because it uses oxygen. The next one is, the last one we'll talk about, is photophosphorylation. And in photophosphorylation, I'm not going to discuss the details of this at all. I'm just going to simply tell you that this forms part of all of the reactions of photosynthesis, but only a part of them. The plants are able to take light and use it because they have these molecules, the pigment molecules of chlorophyll. Chlorophyll is actually able to take the energy of light and put it into electrons. It's actually able to push it into electrons. And we're going to use those electrons, in this case, to make ATP. By the way, it's not the only thing that is made. We're also going to end up making masses and masses of electron carriers. But here, some ATP is manufactured in this process. And that is why it's called photophosphorylation. I'll simply tell you, all of this energy that's produced here is then used to manufacture sugars. So this is a very expensive process, but who cares? Sunlight is free. Okay, and as long as light is shining on these chlorophyll molecules, they can change that light energy into the energy of chemical bonds. So we've got the 3, right? Substrate level, oxidative phosphorylation, and photophosphorylation. All right. So now, with that basis, we're going to talk briefly about metabolic pathways of energy production, because the cell requires energy the whole time. Almost every process that takes place in the cell is going to require energy, no matter what the cell is doing, whether it's building, breaking down, whether it's moving, whatever it's doing, somewhere along the line, we're probably going to be using energy. And the whole point about energy in the cell is that we need a constant source of it, because every time we transform energy, we do so inefficiently. And we lose a certain amount of energy continuously as heat. Put your hand on your arm, feel the heat. For sure, we have evolved to use the heat to keep a constant body temperature, but nonetheless, we still radiate a tremendous amount of heat. And all of that radiated heat is actually waste, because there's no biological system that can use heat to do work. So we need a constant source of energy to replace the energy that we're losing because of the inefficiency of the reactions. So we extract energy from fuel sources. And we do so, the biological systems do so extremely carefully to harvest as much energy as possible. And they do so through metabolic pathways. We've already described what a metabolic pathway is. It's a series of reactions, each reaction governed by a separate enzyme, ending up with some end product and maybe useful products along the way as well. So what we're going to watch and see is that metabolic pathways will strip energy from fuel sources. And the ultimate aim is to put it into a biologically useful form, which is ATP. And all of the things we're going to talk about, we're going to watch as ATP is produced in order to be used particularly in the processes that require an input of energy, such as synthesis of molecules or transport, active transport, all of these things. They all require energy, and they all receive it in the form of ATP, in energy contained in that phosphate bond. A few things to say, just to remind you about metabolic pathways and enzymes. They are stepwise, each step governed by a separate enzyme. Those enzymes can be present in very low quantities because they work very, very quickly, they work very efficiently, and they are used over and over and over and over again. An enzyme is produced, the basis of most enzymes is protein, so that they are produced from recipes lodged in the DNA, from genetic information in the DNA. The recipe for an enzyme is contained in a gene. And in general, it's not entirely true, but in general, you can think of one gene ends up producing one protein, and that protein is very often an enzyme. Okay, so let's look, we're going to discuss first of all how cells acquire energy from one of our principal sources, which is carbohydrate. And you can think, for example, of the simple substance of glucose. What does glucose consist of? Glucose is a 6-carbon sugar, and it has oxygen associated with it, but it also has many, many hydrogen atoms associated with it. And each of those hydrogens attached to the carbons is bound by a covalent bond. So the formula is C6H12O6. So each carbon has 2 hydrogens associated with it. And those hydrogens contain a lot of energy, a tremendous amount of energy, and the cell is going to use the energy of their electrons, but it's also going to use the proton. Remember that the hydrogen atom is comprised of a single proton and a loose electron circuitry. All of the systems we're going to talk about, they're going to strip off that electron and send it off to do work. But in the meantime, they also got these very useful protons in the cell. And we're going to see that the cell is able to use those as well. So how is this energy acquired? Well, it's acquired again through sets of stepwise reactions, metabolic reactions. In prokaryotes, but in eukaryotes as well, but especially in prokaryotes, we recognize that there are actually 2 ways in which cells make use of carbohydrates. 2 principal ways. The first of those systems, when oxygen is available and when an organism is evolved to utilize the oxygen. This is not always the case. Okay? So first of all, I explain that there are many organs, as you know, we are amongst them, that are aerobic. They are able to use oxygen, and in fact, very often have an absolute requirement for oxygen in order to make proper use of the energy contained in fuel substances. These are the processes of respiration. So respiration, these are all very complex metabolic pathways that end up producing masses of ATP and, as byproducts, water. But at the very start of these processes, both of these processes, this one I'll talk about in a second, both of these processes make use of an initial set of reactions here, which is called glycolysis. Glycolysis means literally breaking glucose. And we're going to consider it in terms of just of that, of taking a single glucose molecule and watching what happens. Remembering that there are many molecules in the cell which are made up of polymers of glucose, which can be broken up into individual glucose units, which can also be put through. So first of all, we at like the foundations for all of these systems for utilizing carbohydrates is something like this, is like glycolysis. There are some variations on it, but let's just think for the moment that glycolysis is our basis for the systems of using carbohydrates. There's a reason for that. And that is that this was the first system to have evolved to do so. I just want to check one thing before we go on. Yeah, that's fine. Sorry. Okay, there's a reason why glycolysis is sitting here like the foundation. It's because it was the first set of reactions to have evolved. So these systems initially evolved under completely anaerobic conditions. There was no free oxygen on the planet originally. All of the oxygen was bound to other substances. There was no free oxygen for systems to use. So all living systems at that stage were prokaryotic. They all depended upon this system of glycolysis here, the set of reactions, in order to get their energy, their ATP. This has survived. First of all, the glycolysis has become incorporated into these more complex systems of respiration. But it has also survived because many cells can actually resort to just using glycolysis as their only source of ATP. They have to do so in a special manner, which I'll explain later, which we call fermentation. So fermentation is actually the utilization of this pathway without oxygen. There are some prokaryotes which are absolutely required to do this. They're completely intolerant of oxygen. So they only have fermentative pathways in order to produce their ATP. Those we call the obligate anaerobes. It's an important term to remember, obligate anaerobe. It means it is intolerant of oxygen and must rely on fermentation. But there are many prokaryotes which are facultative anaerobes. That is, they will operate perfectly well in oxygen and use these systems, but they can switch over. If you take away the oxygen from them, they can switch over and they can use fermentation processes in order to generate their ATP. In fact, we can also do that. Eukaryotes very often, depending on the eukaryote and how they're adapted, they can have one or better ability to resort to fermentation. Simpler eukaryotes are better at it than we are. But there is a hint of this in our own cells. In our muscle cells, for example, if we work our muscles so rapidly that we cannot supply oxygen to them, then that is when muscle starts to hurt, when that burn starts. It's when you're starting to deprive the muscles of oxygen. And they are able temporarily to resort to systems similar to fermentation, just temporarily, in order to keep going. Eventually they can't do it anymore. Their muscle cramps up and stops working. But they can do it for a certain amount of time. These fermentation situations, by the way, they produce side product, they produce intermediates, products which may be quite toxic. In our case, we produce lactic acid. Lactic acid builds up in our muscles if we force them to work anaerobically. And that is one of the causes of pain and lingering pain, the stiffness you feel the next day. Part of it is due to the action of lactic acid in the muscles. Okay, so just remember, this system, glycolysis, sits like a foundation. It is incorporated into these elaborate systems of respiration, which we're going to talk about at some length, but it is also present in these systems of fermentation that we will also discuss. Okay, so we're going to talk just briefly about this system. I'm just moving. I don't know if I'm moving for you, but I'm moving for me. We're going to talk about, first of all, about this foundational system, which is the system of glycolysis. And what we're going to see is that in glycolysis, we're going to oxidize glucose. So if we're oxidizing glucose, we're going to be reducing other things. Okay, we're not going to have electrons just flying around the cell. These systems are coupled. So if we're going to oxidize glucose, ultimately, we're going to have to reduce other substances. This is a metabolic pathway, a lengthy metabolic pathway. And in fact, the metabolic pathway divides into two. There are two almost identical portions. The end product of this system of glycolysis is this substance, pyruvic acid. That is something you need to remember. We get in glycolysis, our substrate, initial substrate, is glucose. Our end product is pyruvic acid, along with some other products, ATP being one of them, and various other things as well, as we'll hear. So we recognize in glycolysis that there are actually two phases to glycolysis. The first is called the preparatory phase, and here it is here. I do not expect you to remember all the names of the intermediates. I will tell you exactly when I want you to remember particular names. So here's our glucose here. 1, 2, 3, 4, 5, 6 carbons on the glucose. And in order to get this whole process going, we're actually going to have to put in energy to start it off. And we're going to get that energy from ATP. So this may sound rather strange, because surely we should be manufacturing ATP. Well, we will, but in order to begin the whole process, we need to destabilize the glucose. And to do so, we're going to phosphorylate it. 1, 2, 3, 4, 5, 6 carbons. Here's our ATP. ATP, we're going to split off that one phosphate. Here we produce ADP, and we're going to phosphorylate our glucose there. This produces this, you can see from the name, glucose 6-phosphate. Just remember, it's a glucose phosphate. It's gained its phosphate from there. We're going to do it again. We're going to bring in another ATP. So there we invest another ATP. Again, this is now a completely different substance, fructose diphosphate. So this fructose diphosphate, isomer actually, of glucose diphosphate. Here's 1, 2, 3, 4, 5, 6 still, but they are diphosphate, one phosphate on either end. This is very unstable. And it's enzymatically very easy to break it. And this is exactly what happens. The molecule is actually broken into two parts. And here they are here. Initially, they are slightly different to one another, but they're enzymatically converted into this substance here, glyceraldehyde 3-phosphate. 1, 2, 3 phosphate. But remember, this is really important to follow what's happening. You need to remember that there are two of them, okay? Because we took one glucose molecule, 6, and we split it into 2 molecules of 3. We're only going to, in most diagrams, we're only going to follow one, but remember it's happening twice, okay? It's happening twice. Here's a glyceraldehyde 3-phosphate. Remember that name. You've got to remember glucose. You've got to remember glyceraldehyde 3-phosphate. Here we go here. Now look now comes the point of bringing in our electron carriers, because look what we're going to do. Here's the here's an electron carrier, NAD+. We're going to bring 2 molecules of NAD+. This is because, why? Because we're running this twice. Okay. This applies to each branch. We're going to, here they come here. We're going to strip off electrons and produce NADH. That NADH is very useful. It's very, very useful to the cell because it's an electron carrier and it's carrying hydrogens together with the electrons. In the process, we're going to phosphorylate this glyceraldehyde 3-phosphate. And we're now going to take that, we're going to enzymatically, we're going to strip off that phosphate and we're going to produce ATP. There's our, look how many we're going to make from, remember, 1, 2. This gives you the total. We're going to make 2 ATPs there. So what we've done, we've already paid for these 2. Okay. So any ATP from now on that we make, it's going to be profit in these processes. So here we make 2 ATP. We go on and we go on and we go on. We go on through a whole series of reactions, shifting this phosphate around all over the place until ultimately we strip those phosphates off, add them to ADP again, and we end up making another 2 ATP. So what we did in the preparatory stage, we invested 2 ATPs. We ended up producing 2 molecules of glyceraldehyde 3-phosphate. We took each of those glyceraldehyde 3-phosphates and we pushed them through a series of reactions. Each glyceraldehyde 3-phosphate made a molecule of ATP there and a molecule of ATP there. In total, we made 2 molecules of ATP there, 2 molecules of ATP there. We made 4 in total. And we've invested 2, we made 4 ATPs. We have now a profit of 2 ATPs. It's not a lot, but it's a profit. And many organisms are able to survive just on that. They are able to survive with that in order to do so, their metabolism slows down tremendously. These anaerobic systems that only use glycolysis usually work slowly. They're not as efficient, but nonetheless, it is good enough to keep many organisms going. The end product you must also remember, and it is this pyruvic acid. And you'll see why I want you to remember it, because this is now the substrate, which is going to be taken off into all of the other pathways that we're going to talk about. Okay, so that 2 molecules of pyruvic acid are now produced at the end of glycolysis. Here's the summary here. The profits that we reap from the system are, first of all, the net 2 ATPs that we got, but very importantly, these molecules here of NADH, 2 molecules of NADH are produced. And that NADH is carrying those hydrogens together with these very, very energetic electrons. In fact, this here is an essential step in the whole process here. If something happens and we stop this from operating, this reduction of NAD+, then these reactions cannot carry on. And I'm emphasizing that because we're going to see that this becomes important in those organisms which do just rely on glycolysis. They start using up all of their NAD+, and they have to do something about that situation. Okay, in summary, we're going to take a glucose molecule with its 6 carbons. We're going to biphosphorylate it. We're going to phosphorylate it twice. It's going to have 2 phosphate groups attached. Makes it very unstable. It's enzymatically, it's very easily broken into 2 molecules of glyceraldehyde 3-phosphate. Each glyceraldehyde 3-phosphate proceeds through a series of, through a metabolic pathway to ultimately make pyruvic acid. That's a 3-carbon sugar, a 3-carbon compound, I'm sorry, pyruvic acid. It has no phosphates attached to it. What happened to our 2 phosphates here that were in our 2 molecules of glyceraldehyde 3-phosphate? They got added to ADPs here. They got added to ADPs, so we ended up making 4 ATPs for an investment of 2. We had a profit of 2 ATPs. And along the way, we made 2 highly energetic electron carriers of NADH. Okay, so I'm going to stop there for today. A little bit shorter than usual, but you have a little bit of extra time to study for your test on Wednesday. Okay, and I will see you. I won't see you on Wednesday. I will send you an announcement reminding you of how to access your test. Okay,